plant tissue culture lab, Paralakhemundi campus. This is our culture, overall tissue culture lab. This is the units we are used for the media preparation unit. This is one block, and we are maintaining pH meter, plant exterior, and weighing balance, hot air oven, and also culture bottles. We are sterile culture bottles we are keeping inside this, and we are weighing the materials required for media preparation. We are making sugar and agar, and also any other antioxidants are required. We have to weigh in this balance. This is agar, and also distilled water. Double distilled water we are bringing, and you can go for the autoclaving. The next step is autoclaving. Autoclave, we are going to set autoclave at 121 degrees centigrade and 50 lps pressure. These are the culture bottles after autoclaving of this decontamination. Then you can go for again dipping in potassium dichromate solution and at least overnight 5 to 6 hours. After that we are going for again autoclaving in the autoclave sterile bottles up to 121 degrees centigrade for 25 lps. After that you are going for store in the oven condition. Then you can use for further culture of the bottles. After that you can go for the media preparation. Then preparation, we can store these bottles into the culture room. This is the laminar airflow, and culture room we have to maintain the sterile in the condition we are creating by UV light at least 15 minutes UV sterilization. You have to go for this laminar airflow. The wind speed is 12 kilometers, 1.2 kilometers per hour. We have to maintain. After that, you can sterile with 70% ethanol. We can go for the culturing of the material. Okay. Inoculation process, you have to go for that. In the culture room, this is a growth chamber. Here we are maintaining cultures. You see the cultures preparation we are doing here. The plants, the tissue culture plants we are growing. She is our lab in charge. She is taking care of everything when sterile conditions. She is making this, and also components also she is distributing to others on the register entry. Further process detailedly explained by Ms. Saraswati. She is the lab in charge of this tissue culture lab. Okay, proceed.
Greetings of the day. I am Saraswati Pati. I am the technical in charge of the tissue culture lab. Here our faculty in charge is Dr. Gautam Kumar Das. Here we are doing the different tissue culture program from different type of plants and different type of crops. Basically, we are doing the arity-listed forestry species. We know the arity-listed forestry species are the red sandalwood, ochar, every different type. But we are doing the red sandalwood, basically Pterocarpus santalinus. After that, we are also developing the protocol of the red sandalwood and standardize the protocol of red sandalwood and chrysanthemum plants, flowering plants. Now we are doing the different type of medicinal plant also, like stevia, and some vegetable crop like brinjals. This is our lab, and different type of instruments also there, like pH, weighing balance, autoclave, hot air oven, and the culture room and the laminar air room. Inside our culture room, we have to maintain the temperature, the humidity. We know the temperature and humidity. After that, we are doing the tissue culture program.